There is absolutely no time for break here. So let's go on with the next session. And I will myself give the first talk. So let's see how this will work. So I'm from the Tata Biocenter, and I will just give you a brief introduction of who we are. So the Tata Biocenters, there are two of them, one in Sweden and one in Czech Republic, and we do have offices in California and in Germany. We are essentially a training provider in molecular diagnostics and in particular in real-time PCR. We are also one of Europe's largest service provider for companies, hospitals, and assay design, validation, and analysis of molecular samples. And essentially the reason that how we have developed is that we work very, very close with essentially all the instrument providers in the field. So our labs are very well equipped. We have over 20 different real-time PCR instruments, robotics, and you name it, all systems for designing experiments and analyzing data. And we also welcome students and postdocs to come to our lab and use it as a core facility. It's very attractive, and you are supported, and through us you are supported by the instrument manufacturers. So this is, we also have our own product development. In fact, quite a few of the products we use today were in somehow co-developed or validated by us because we work very close with many of the leading companies. We are very keen on quality control because one of the issues, and I think it has been realized also during this conference and pointed out by several of the previous speakers, it's quality. We are doing quite challenging analysis, and it's easy to go wrong. And particularly many inexperienced labs get quite frequently the impression, particularly perhaps from the sales reps, that things are much easier than they are in the real life. And essentially a couple of years back, we, together with some colleagues, a few of them are here, and Michael, I believe tomorrow will be talking only about that. We presented essentially guidelines or recommendations in what you should test in order to validate that the results you generate are reliable, and also that information should be provided when you submit papers for publication. And today, I think it's about 20 journals actually request the data are compliant with the MIKE guidelines to be accepted for publication. This was just put in Nature, and there were a couple of stories there about problems caused by poor qPCR data. We are also involved in a quite important European effort. I don't know if somebody will be talking about it here. Hopefully, there was a talk about that yesterday in Barcelona. It's a European effort aiming to standardize the pre-analytical process. That is what happens from the point of sampling until the actual analysis. And with a number of colleagues, we did ring trials. Samples were collected in Florence, in Italy. Maybe that's why Mario is not here, because they took all his blood, I know. And then they distributed the blood samples across some 200 laboratories in Europe and asked them to do something that may sound trivial, just purify the RNA and then send it back, and the Florence lab tested the quality. And in a sense, what they found was that about 30% of European routine laboratories have serious issues purifying RNA from blood. That's quite serious because these labs are delivering reports to the clinicians, and based on that, decisions are made. We are a certified lab by the ISO, and the Speedia project attracted the interest of ISO, and now there is a working group that is drafting a new set of guidelines for the pre-analytical process, and we expect those to be in force end of this year. Anyhow, that was a brief introduction, so let's talk about single cells. Well, the first and most important question is, of course, why do we do single cell? Why bother? And I think it's illustrated quite neatly by this graph here showing the islet of Langerhans, which is stained for the delta cells. This is the same islet stained for alpha cells, and here we have the islet stained also for beta cells. These cells, that's three very, very different cell types. They respond to different stimuli, they express different genes, and of course, if you analyze the entire islet, or even if you cut a piece of it, you will get the collective response of all the cells because the islet is a mosaic. If we really want to understand the response of the different cell types, we really need to go down to the single cell level. And that's essentially what attracted us. And actually, so many years ago, one of my students actually visited Birgit Liss, who gave a talk earlier. She was in Oxford then doing postdoc. And together, essentially, they started doing single cell work. So these data are pretty old. But what we did in our lab, using essentially the patch clamp technique to aspirate a cytoplasm from the cells, we analyzed a large number of individual cells. These are beta cells, and this is also cell line, Min6 cell line, for the expression, in this case, insulin 1. We got brilliant data, but we also found a great heterogeneity. There's a very, very large variation among the number of transcripts, insulin transcripts, among the different beta cells in that cell line. And that also turned out to be the case when we isolated them from islets of Langerhans. So, and we are a little puzzled. This is a graph just showing some of the statistics. It's a frequency diagram, essentially showing that out of the cells we analyzed, this happens to be beta-actin expression instead. 40 cells had between 0 and 100 transcript, 25 had 100 and 200 transcripts, some 14, 200 and 300, and so forth. What is striking is that most cells have very, very few transcripts. And then there are a few cells that have a very, very large number of transcripts. Taking these data and just plotted them with another scale, same data as in the previous graph. The difference is that this scale is now log scale, showing the log of beta-actin. When the data are plotted in log scale, you see that they can actually be modeled quite neatly with a normal distribution. And essentially this indicates that transcripts among like cells show a log normal distribution. And since our finding almost 10 years ago, this has been shown for virtually any transcript in any kind of cell. So this seems to be more or less a general phenomenon of cells. I know actually of only one exception myself, and that's oocytes, because they don't have any RNA metabolism. There may be more, but this is what we usually observe. And it does have a number of consequences, because if you're interested in, for example, determining how many transcripts do we have in a typical cell of a population? How do we do that? Well, if we study a regular normal sample containing 1 million cells, we measure the total amount of transcripts and we divide by the number of cells. Well, this is how we calculate the average number of transcripts per cell. But this is known as the arithmetic average, and that number will only be the number of transcripts in a typical cell if the underlying distribution is normal in the regular linear scale. But that's not the case for cells. Because of the log normal distribution we observed, the number of transcripts in the typical cell is actually given by the geometric average. And unfortunately, that cannot be determined in a classical experiment. The only way to determine the geometric average, that is the number of transcripts in a typical cell, is by performing single-cell profiling. I guess you found that kind of confusing. Well, you are not alone. This is how nature summarized our findings, so we confused some more people. Anyhow, maybe it will work from here. So, of course, the question is then, what is the reason? What is the reason that we do have this log normal distribution? Well, we know that, but those data were not, that was not demonstrated by us. It was several other groups. These data are from Jonathan Chubb's lab, who was one of the first that did those studies. And I will actually show it showing a movie from his lab. Hopefully it will start. Yes, it does start. What you see here is a cell, and fluorescence in situ hybridization is used to visualize the transcripts. And what you see is as a movie over time, and you see that the intensity goes up and then it disappears again, clearly showing that the number of transcripts in the live cell changes over time. It's not constant. And from these observations, it was seen that this is roughly how the number of transcripts vary over time. We have bursts. Suddenly, the number of transcripts in the cell increases rapidly, and then they decay. And then you have a new burst and a decay, and so forth. And if you integrate these bursts, you end up with a distribution that actually is a gamma distribution, but it can be pretty neatly approximated with the log normal distribution, which is essentially what we observed in our experiments. The time scale of these bursts, they seem to depend on, they vary with cell type, they depend on the gene, but they seem to be of the order of 10 minutes to perhaps an hour at most, and likewise, that's the duration of the pulses. Notably, our colleagues doing similar kind of experiments in protein expression, they also find burst kinetics and log normal distribution for proteins. So it's nothing that is seen only for mRNA. It's absolutely seen for proteins as well. Notably, the time scales, at least as reported in literature today, are much longer for proteins. The pulse intervals seems to be hours up to perhaps days. The reason they are different, we don't know yet. Even in our first study, we could measure more than one gene per cell. We measured 5 transcripts per cell. They are shown here, insulin 1, insulin 2, and we measured 1 reference gene or potential reference genes. We don't use reference genes today. We didn't know that at that time, and 2 ion channels. And if we measure 5 genes per cell, we can do a quite interesting analysis because we can look at the correlation. We can test if the genes are expressed at the same time in the same cell. And the correlation is calculated by calculating a correlation coefficient, which is a number between -1 and 1. A value close to 1 indicates that the 2 genes are expressed at the same time in the same cell. A value around 0 means they are expressed independently of each other, and a negative value would indicate that when one gene is expressed, the other one is not. Looking at the data, we only found 1 high correlation, and that correlation was very high. Notably, it's a correlation between the expression of insulin 1 and insulin 2. This is mouse model. Mice have 2 insulin genes. This shows that in the beta cells, the 2 insulin genes are transcribed at the same time, so the bursts are correlated. Interestingly, the 2 insulin genes are located at 2 different chromosomes. So there must be a mechanism for the cell to synchronize the transcription of genes on independent chromosomes. There is quite some data today indicating the nature of these mechanisms, suggesting it's actually a localization process, but that's, again, separate. Anyhow, it was nice measuring 5 genes per beta cell, but we wanted to move forward. And then together in several collaborations, we worked out the workflow that, at least in our hands, is pretty robust today. Essentially, starting with cell collection. We have had a few slides earlier today about the different methods. Today, we mainly use FACS. We have developed a reagent for direct lysis, which means the cell is dropped into a test tube with this lysis buffer. The cell is lysed, and the reagent is compatible with RT-PCR, so we don't need to do any washing, which means that there are no losses in the workflow. We have a highly optimized reverse transcription reaction, and then we perform preamplification. The reason we perform preamplification is that if you want to analyze many genes per cell, well, we have a problem, at least if you want to do that in a singleplex measurements, because then we have to take the content from a single cell and divide it into, let's say, 100 aliquots to analyze each aliquot for one transcript. If, in order to have a reliable measurement, QPC, or actually any kind of measurement, starting with, from a test tube, that test tube actually has to contain at least 35 target molecules, roughly. And the reason is that if it contains less, then actually the noise due to Poisson distribution, sampling noise, gets too high. It has nothing to do with the technique. It's the sampling that will cause such high noise. So if you want to analyze 100 transcripts starting from a single cell, and you need to divide it into 100 aliquots, well, you wouldn't need to have 35 times 100, that is 3,500 transcripts to start with. And most cells won't have that, if nothing else because of the underlying log normal distribution. For most transcripts, almost all transcripts, most of the cells will not have that many, will not have not that many copies. So therefore, what we do is that we introduce a preamplification step. In our hands, for single cell work, it turned out that a parallel PCR worked best. So essentially, we mix primers for the targets we are interested in. We run PCR, but we run it for a limited number of cycles only to make sure the reagents or the reactions do not compete for the reagents. It's very, very important the preamplification works well and it's validated. One of the most critical things is to have highly, highly optimized qPCR assays. We actually spend a lot of time optimizing our assays. This is just a plot showing how we typically validate the performance of the assays. Then we also validate the preamp, and that's also pretty straightforward. Essentially what we do is that we start with a test sample, typically a representative cDNA sample, and we analyze the samples either directly or via preamp. We do it for all the transcripts, and then we compare the CQ values or the amount that we measure with and without preamp. And in a sense, if the preamp works fine and there is no bias in it, we would expect all the difference bars to be at the same level. They are not. Some are off, like here, 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 and that indicates that there's a bias for those assays. However, a bias is actually not particularly serious in those kind of analysis because it will cancel when we do the calculations. In fact, we are already relying on the bias not being important because we are not, none of us, I would say, is really correcting for a bias in the reverse transcription. And our lab has studied that and published it. The yields in the reverse transcription can actually vary more than 200-fold among the different transcripts. 200-fold, not 200%, 200-fold. However, it doesn't matter as long as things are reproducible, because when we are comparing samples, the bias will cancel. So that's not important. So what really matters is whether we have a reproducibility here. And the reproducibility is calculated from the replicate. So we can absolutely test for reproducibility. And doing that, it's actually only one single assay here that had to be trashed or redesigned, actually, in this case. In order to get statistics, you have to analyze a pretty large number of cells, typically hundreds or more. And if you analyze hundreds of transcripts, well, that's many qPCR reactions, and you have to work with high throughput platforms. We use either the QuantStudio from LifeTech or Thermo today, or the Biomark from Fluidigm. A speaker earlier this morning pointed out that there may be an issue with genomic DNA, so I actually added this slide here. I think it's quite illustrative. And one point I wanted to say is that actually, even though many company representatives would tell you that you avoid genomic DNA contamination by designing assays across introns, that is usually not true. And the reason is that about 15% of human genes have pseudogenes. And about 50% of the pseudogenes they don't have introns, because the pseudogenes are copies of transcripts formed by retrotransposons. And those are amplified even with intron-spanning primers. You can't avoid that. And there may be many pseudogenes per active gene. GAPDH, for example, in humans is present in 70 copies, the pseudogene. In mice, I think it's over 300. So there may be a significant genomic DNA background, even in single cells. As Birgit Liss said earlier this morning, one way to test or control for genomic DNA background is to do RT negative controls. You can absolutely do that according to the MIKE guidelines. The RT negative should be 5 cycles above the positive sample. That's great, but it's expensive, particularly if you want to run 10,000 reactions as we do. That bothers us. So we developed a strategy how to compensate for genomic DNA background and keep the cost down. And this we do using an assay we call valid prime. A valid prime is an assay that amplifies specifically the genomic DNA, but it's not targeting a gene. So it's not amplifying cDNA. It targets a genomic sequence that is conserved and present in exactly 1 copy per genome. And then all samples are analyzed with a valid prime. So we are essentially measuring genomic DNA background. That is not sufficient for compensation, but the valid prime kit also contains a genomic DNA standard. So all our assays are then tested on the genomic DNA standard, and this will tell us how sensitive are the assays, that is your assays, the assays you have developed for genomic DNA. And this will depend on whether you have intron-spanning primers or not, or and also the number of pseudogenes. You don't know, you just measure it. And from those extra measurements you can compensate for the genomic DNA background in all your samples. For a large study, you save 95% of the cost because you measure M plus N controls instead of M times N controls, which is necessary in regular RT negatives. Oh, that was just a comment. This is published, and this is how you compensate for it. Then we use multivariate methods for analysis, typically in pathway analysis. I will show you an example. The system I will be describing now is a study of astrocytes, and we use mice models, and the astrocytes express green fluorescent proteins. We can collect the astrocytes from mice brain by faxing. And what we have done here, or rather our collaborators have done, is that they've induced focal cerebral ischemia, that is brain damage, and we are monitoring how the astrocytes are reacting to that, essentially how the brain is trying to repair the damage. These are just some data plotting the frequency diagrams showing the number of transcripts that we have in different cells. As before, you see that they have a kind of log normal distribution. It's always log scale here. Or, well, if you look closer, not here. This is not a log normal distribution. There are 2 peaks here. And if we enlarge that, you can clearly see that there are actually 2 different log normal distributions. This happened to be Wymington. What this tells us is that this population of astrocytes is not homogeneous. There must be at least 2 different, I will use the word types because I don't, but I will be a little careful with that because we have not defined cell types, if you recall. It's difficult. But anyhow, there are 2 different groups of astrocytes here. And when you have 2 different groups of cells, you can start comparing them. You can do statistics, t-test, plot differential expression. You can do nice volcanoplots. Great, but not particularly useful. And the reason is that, first of all, if we measure the expression of a very large number of genes, then the statistics will be shaky because there will be very large risk of having false positives there accidentally. Secondly, we don't expect the genes to be activated independently of each other because that's not how biology works. And if we analyze data with using a regular t-test, we are analyzing the genes one by one. And in fact, we do assume they are independent of each other, which they are not. There are much more powerful methods to analyze data that actually exploit correlation. And one of the most powerful, we saw it earlier, is principal component analysis. And I will illustrate how that works. Okay, what I do is that I will do a dynamic PCA, because I need to do it on the computer because it's a three-dimensional plot. So this plot here shows the astrocytes. So every ring here that's actually one cell, but it's a plot. So it's a drawn spheres, and the spheres are positioned in a three-dimensional coordinate system reflecting the expression of the genes. We have seen these PCA plots before, but what is important when you do this kind of analysis is that typically we are measuring the activation here of astrocytes as a function of time, and the color coding shows time. So blue are astrocytes collected just after the induction of, before actually the induction of brain trauma, time 0. Green are 3 days after trauma, yellow 7 days after trauma, and red or 14 days after trauma. The axis, or mathematical linear combination of the expression of the different genes that separates or really emphasize the time development. And the analysis here is based on 47 genes. But what actually happens is that most of the genes are not really important because they are not responding to the trauma. And therefore one can use a filter, essentially a filter based on variance. So what I'm doing now is that I'm removing genes from the analysis. Genes that do not change expression over time. And by doing that, I will actually improve the PCA analysis. And if I can do that quickly just now interactively, you see that there is a perfect separation between the red and the rest. And the red are reactive astrocytes. That is how they have changed expression during the 14 days after the trauma. And furthermore, if I use a slider here to actually indicate the clusters even more clear, you see that there are actually two types of reactive astrocytes. Not only that I do change the profiles of the astrocytes, I get two different types because of or in response to the trauma. And those tools are really handy because you do improve the analysis, or rather the resolution greatly by essentially throwing away the genes that are not contributing. And this is just a plot indicating the genes that became active as a function of time during the activation of the astrocytes. It's the same data as before, it's just shown in a two-dimensional plot. Correlation. So what we do here is the same thing as we did for the beta cells, but now we have 47 genes. We're looking at correlations, looking for genes that are active at the same time in the same cell. And the correlation is indicated in green. Dark green means that the correlation is significant at 99% confidence, 95 is light green. And you see there are blocks. This is just one set of all the data. And the blocks indicates then genes that show correlated expression. And these genes are then expected, I would think at least, to be part of the same expression networks or pathways, because when we have a stimuli here, well, in this case brain trauma, they are activated, but they are activated synchronously. However, one has to be careful with just interpreting the correlation coefficients. And this is illustrated here because if we have 3 genes, it could be that there's a master gene here that somehow triggers the expression of 2 other genes. And that will lead to an indirect correlation between these two genes, although they don't have any direct interaction. It could be that gene A somehow triggers gene B, which triggers gene C, or there may be a correlation between, or they may be interacting all of those. To illustrate the phenomenon, or to exemplify that, I will use actually an analogy. I will use an analogy actually from a Swedish newspaper some 20, 25 years ago. It was a journalist in Sweden, and he became interested in how hot weather changes what we do during summer. And he found that during summer, when the weather is hot, we eat more ice cream. Okay? And then he also looked at the statistics of drowning, and he actually found when the weather is hot, there are more drowning accidents. So he published on the first page in Swedish newspaper a theory that eating ice cream causes drowning. Okay? All right. I don't think we believe in that, but there was obviously a correlation here. And this is the same thing when we analyze the genes. If we really want to find the mechanism behind it, we have to get rid of those accidental or indirect correlations. And that can be done by a so-called partial correlation analysis. Essentially, if we know these are correlated, we can easily calculate the correlation we expect to be here, then we subtract it and see if there's anything significant left. That's how it's done. And doing that, we could identify expression pathways or networks of genes that become activated in the same cell, in the same astrocyte cell, of course, in this case, as response of trauma. And this is pretty cool. Pathway analysis is hot today. It's because a lot of pharma companies are interested in that, and there are many groups doing enormous efforts in statistics on that. And they are struggling with enormous data sets, because if you work with classical samples and you apply a stimuli, you will be affecting thousands, 10,000s of genes. Here, on single cell level, everything breaks down in much smaller pieces of information. It's so much easier to identify expression pathway on single cell level, because we only see those that are active in the same cell. Well, this was just brought up in science. Anyhow, can we go beyond a single cell, by the way? I mean, I know this is a single cell conference, or maybe it's not, but I hope I am allowed to go beyond a single cell. This is a single cell. It's a big cell. It's an oocyte. The nice thing about oocyte, this is an oocyte from a frog, is that the two halves of the oocyte have different stain, which means that I can take the oocyte and embed it and cut it in a cryostat. And if I do that, so I slice the oocyte, I can measure intracellular mRNA profiles. And that's what we have here. So these are mRNA profiles from one side of the cell to the other. And look how they agree for the different transcripts. The transcripts, they're not in the middle of the cell, by the way. They're a little bit towards the animal pole. And in fact, that's where we expect the cell the nuclei to be, because it's not perfectly in the middle. But look, we found another group of transcripts on the other side of the cell. They're here. And notably, we found a few transcripts more or less exclusively in the last 15th segment of the cell. So these transcripts have to be almost associated with a cell membrane. I can skip. We just validated it with digital PCR, but I will not. This is just a digital PCR validation. But since we are short of time, I will not talk about digital PCR. But anyhow, it has a few consequences related to cell division that are quite interesting. This is the oocyte, and we know how it divides. That has been known for a long time. This is how it divides. And the gradient that we are measuring is along that line. So, the two daughter cells here are not affected by the gradient. They can still be identical. This is the second cell division. Still, the four blastomers can be identical because this is the gradient. But at the third cell division, they will be different. But the nice, cool thing about it is that this was predestined. The asymmetry was there already in the original oocyte. It planned to divide asymmetrically. It's a clever guy, huh? These are just showing some raw data measured on the Fluidigm instrument. They are nice. 10,000 data curves here. You can spend quite some time looking at those. But why do I show raw data? What's special about those? I mean, there are, I think, close to 1,000 biomarker instruments out there today. The special thing about these data is we are not measuring mRNA. We are not measuring microRNA, not DNA. We are measuring proteins. So how are we doing that? Well, we are doing that using a technology developed at the Swedish company Olink. And what they have done is that they have they've identified pairs of antibodies that have been tagged with DNA oligomers. And the oligomers are complementary at the 3' ends. And they are complementary such that if they get a chance to hybridize, they will actually prime a PCR reaction. So we can form a PCR product and detect it by qPCR. But they will only come close if they are bound to protein, because the binding of the two antibodies brings them in proximity. And today, together with Olink, we have panels of 96 assays measuring protein expression. Essentially, we analyze 1 microliter of crude sample, such as serum, plasma, lysate. And there is a panel for oncology, one for cardiovascular, and there is one for inflammation coming out. And since I heard David was here, I have to show these data as well. These data were generated by a colleague of mine, Anders Stålberg, together with David here. And essentially what they did was that they took a single cell, they divided that single cell into a few aliquots, and the aliquots were analyzed for RNA separately, DNA, and protein. So this is the first time mRNA, microRNA, long non-coding RNA, DNA, and protein was assayed in the same cell. And that, of course, opens for the possibility to look at correlations between DNA, RNA, and microRNA levels in the same cell. The reason DNA varies, I should stress that, is that this is a construct. So we use plasmid that we're coding for a transcript to essentially activate everything. So therefore, we do have a correlation with the DNA level. And indeed, we found pretty nice correlations between DNA, mRNA, and protein levels in the cell. We found also some positive correlations with certain microRNAs. And notably, for the first time, we actually found a negative correlation. We don't really understand the biology behind it, but it's quite interesting. And I'm really not surprised that we should have negative correlation. There will be. I mean, one gene may suppress the expression of another gene, although this is a negative correlation was actually the DNA plasmid, but that's another thing. Anyhow, that's what I wanted to share with you. These are some of our collaborators and partners, and I'd be happy to take your questions.
Thank you. One question?
I have a question relative to the Olink technology you just showed us. Can you comment the stability of the DNA, so single-stranded DNA attached to the antibodies? Is it stable? How stable it is?
We've never lost it. No, I mean, no.
To lose it, I have no doubt. It's more degradation.
No. Well, it's very stable. There are actually a few different ways how to conjugate DNAs to antibodies. They have developed over time. And in fact, before starting working with Olink, we were working with immuno-PCR, so we have been doing DNA conjugation to antibodies for quite a long time. If you're unlucky, you may be attaching the DNA to a part of the antibody that will actually interfere with the binding of the antibody. That absolutely happens. You have to validate it. But once you have conjugated it and once you have purified it, I can tell you at least some of the constructs we have done or we manufactured for, let's say, 7 years ago, they still work very well. But whether that holds for all antibodies and all oligos, I can't really tell. But we have very good experience out of stability of these constructs.
Maybe you could explain a little bit more, for my naive understanding, the advantages of your pathway analysis by correlation. Usually when people do these pathway analysis, they start by generating differential gene lists, and then they look into these GCEA gene banks and check whether this is enriched. Now, what would be the advantage of your approach?
I was not fully clear on that. Actually, when I said that we typically use pathway analysis, we do, but I didn't refer to the correlation analysis, pathway analysis. The pathway analysis we do is typically going to Ingenuity database and trying to identify or correlate with the known pathways. However, the correlation analysis we do is I would rather call it a network analysis, because it's rather a network. The genes do not necessarily have to be in the same pathway, but I should say that currently we don't really know what causes the correlation, because the only thing we know is the correlation means that the genes are on at the same time at the same cell, and they are not constituently expressed. None of these genes are. Most of the cells are blank, and then suddenly this cell has a lot of transcript, but it happens to have also the other transcript there. So there must be some underlying interaction there. But I wouldn't really call it traditional pathway analysis, because I think rather we are breaking some new ground here. I know there are a few statistical groups out there that have started to become interested in that. We are not really experts more than we use pretty basic statistics like correlation analysis and partial correlation analysis. And the networks we draw, it's actually very simple. It's just connectivities are drawn when the correlation after compensation for the indirect correlation is significant. That's essentially how we draw them. So it's really no big science behind it. It's pretty straightforward, but it seems very, very powerful. That's the thing.
Thank you.